Hello everyone. Today's cell organelle is Golgi apparatus. Golgi apparatus can be seen with both light microscope as well as electron microscope. It was discovered by Camillo Golgi in 1898. Location: Golgi apparatus, they are found in all the eukaryotic cells, but they are absent in prokaryotic cells. They are excessive in the secretory cells, like the sieve tubes of plants and uh, the sperm cells of bryophytes and pteridophytes. Although they are present in all the eukaryotic cells, but the Golgi apparatus, they are absent in RBCs. Now, the structure of Golgi apparatus, they vary in size and form in different cell types. As seen through electron microscope, the Golgi apparatus, they consist of parallel, flat, tube-like structures which are intercommunicating, and these are called cisterns. The cisterns, they vary in number in a cell. Their number may be 3 to 7 in the animal cell, but their number may be 10 to 20 in the plant cells. They are equally spaced and piled up, and the space between the cisternae of Golgi apparatus is called intercisternal cytoplasm. This is the intercisternal cytoplasm. The cisternae may be flat. The cisterns, they may be flat, but they are often curved and they have a distinct polarity. The two poles of Golgi apparatus, they are called, this is the cis phase and this phase is the trans phase. The cis phase, it acts as the receiving. The cis phase is the receiving end and the trans phase is the shipping department or the departure end. The cisterns, these cisterns, they are free of ribosomes and they usually have the swollen ends. The cisterns, they are free of ribosomes and they have the swollen ends. These are the swollen ends of the cisterns. Now, these swollen ends, they suggest that and cisterns, because they look like smooth endoplasmic reticulum, it is believed that the Golgi apparatus, it is formed from endoplasmic reticulum. Now, the trans phase of Golgi complex, it arises from secretory vesicles. The second structural component of Golgi apparatus is tubule. Tubules, they arise from periphery of the cisterns as a tube-like structure. They arise from the periphery of the cisterns as tube-like structures, and some of these, they enlarge to form the vesicles also. These are the third structural component of Golgi apparatus. These are the vesicles, and these vesicles, they pinch off. They get separated from the tubules of the cistern and they lie towards the trans phase of the Golgi apparatus. Now, the chemical composition, the cell membrane of the Golgi apparatus, it is the same. It is the phospholipid bilayer which is sandwiched between the proteins. Now, what are the functions of the Golgi apparatus? It helps in packaging of materials, synthesis of carbohydrates, formation of glycoproteins, formation of lipoproteins, and formation of cell wall. It helps in formation of lysosomes, formation of yolk, etc. Now, if we look at the origin of Golgi apparatus, in the nucleus, it was bounded by the nuclear membrane. From the as the extension of nuclear membrane, it was formed the endoplasmic reticulum. This is the nuclear envelope, and the extension of nuclear envelope, it gives off the rough endoplasmic reticulum, and from rough endoplasmic reticulum is formed smooth endoplasmic reticulum. This smooth endoplasmic reticulum, it gives rise to Golgi apparatus, and the ends of the Golgi apparatus, the vesicles, they give rise to the lysosomes. The formation of organelles is interconnected. Now the lysosomes, they form primary lysosomes, they form secondary lysosomes and tertiary lysosomes, and ultimately they form the residual bodies. And these residual bodies, they go to the cell membrane, and they help in excretion of excess of materials or excess of water present inside the cell.